Hello everyone, welcome back to the session. So in yesterday's class we have discussed the complete information about cell cycle, and in today's class we are going to discuss about the cell division. Okay, so we have started the cell biology chapter and today is the end of the class, which means end of the cell biology, and from tomorrow we are going to start a new chapter, and the last final session will be the cell division. See here, so in yesterday's class we have left some topic called this P53 and P21, and we are going to discuss about that, and we are going to discuss about the cell division completely. And what is cell division? So what do we have discussed in the previous class? So in the cell cycle we are having G1 phase, S phase, G2 phase and M phase, and this M phase, the complete cell division has been taking place in the M phase, and we are going to discuss about that M phase completely here. So how can we define the word cell division? Here one cell is going to divide 2 cell or 4 cell. How? Okay, in what way one cell is divided into different daughter cells? See here, the cell division is a process by which a parent cell divides to form a daughter cells. And why one cell has to divide into daughter cells, and what are all the essential characters of this cell division? See here, the first one will be the cell and growth and development. So if a particular cell attains a proper size, and if it attains a growth, it has to divide, and there is a repair and regeneration. So we have discussed if there is any damaged I got in my hand, so after that cells it will be dead. That cells present in that particular area has been dead. Now that dead cells has to be replaced by newer ones, and for that process cell division is mandatory. So that is what repair as well as the regeneration process, the cell division is very important. Now the reproduction. So the same thing, the cell has to reproduce. For example, the lifespan of RBC is 120 days, and if 120 days if the lifespan of this RBC is 120 days, after 120 days no RBC will be present. And is that possible? Without RBC our cell life will be smooth? No. See here, if after 120 days the cell has to reproduce, so only then the newer RB cells has been produced. So like in the same way, all the cells have a particular lifespan, and that has to be reproduced to produce a new cells. So that is why reproduction is very important. And maintenance of chromosome number. See here, the chromosome number, in the sense the total number of chromosome will be 46, and that 46, and even after the cell, see for say for an example, say this is one cell, and this cell is undergoing a cell division process, and as a result of division it divides into 2 daughter cells, and in these daughter cells again the 46 chromosome has been divided into 2. So we cannot say the 2 daughter nucleus have 23 and 23 chromosomes. No, that is what we have discussed in the yesterday's class itself. The chromosome number and the chromosome content has been divided in the S phase itself. Just see here, and so the mother cell has 46 chromosome, and after division the 2 daughter cells is also having the 46 chromosome number. And genetic continuity. So the genetic continuity, in the sense the parent DNA is the one undergoing a replication process. So that is what we are saying, that how the parent cell, the 2 daughter cells will look like the same exactly how the mother cell will be. So this is what genetic continuity. So these are all the things, these are all the factors which is essential for the cell division. Now, and we are having different types of cell division. So as of now, just know the cell division is just division of a cell, and as a result of division one cell is divided into 2 different cells. And in the type of cell division you should know how the cell has been divided into 2 cells, and in that division we are having 3 different types of division, but majorly 2. But in a primitive individual we can see the amitotic cell division. Just know that amitosis is a type of a cell division, and this amitosis is also called as a direct division. We are going to discuss about that, what is amitosis and why we are calling them as the direct division. And just know this mitosis is also called as a equational division, and the meiosis is called as a reduction division. So just know As of now, just know that we are having three types of cell division: amitosis division, mitosis and meiosis, and the other name of these types of cell division. And the first division will be, we'll discuss this slide a little bit later. Now, the first type will be the amitosis cell division. See here, amitosis in the sense a simple division. So this amitosis is called as a simple division. As it is a simple division, it is seen in the simpler organism or primitive organism. And see here, this amitosis is a simple cell division where a nucleus divides without spindle formation, without distinct stages. So in yesterday's class we have discussed there what will be the importance of the spindle fibers, and also we have studied about the spindle fibers in the microtubules also while we are discussing about the cytoskeleton. Just know here, whenever the cell has been divided into two nucleus, definitely there will be the involvement of this spindle fibers. But in amitosis, there will be no importance of the spindle formation, and there will be no distinct stages has been undergone by this parent cell. So that is why we are saying this particular simple division as an amitosis cell division. Just look at the picture. This is a parent cell, and there is an elongation of a cell as well as the nucleus, and as a result that the nucleus has been divided into many nuclei, and each nuclei has been surrounded by a cytoplasm, and immediately after the division it will be divided into two different cells. Okay, so this is what an amitosis division. So as the cell has been directly divided from the parent cell without any sequential stages, we are calling this particular amitosis as a direct cell division. Now, what are all the key features and what will be the difference between this amitosis with the mitosis and meiosis? What will be the crucial differences? Just look at this. There will be no spindle fibers. Spindle fibers is very important in case of mitosis and meiosis, but there will be no involvement of spindle fibers in case of amitosis. And no chromosome condensation. Chromosome condensation is also another significant process taking place in the prophase of both mitosis as well as the meiosis, but there will be no chromosome condensation in case of amitosis. And the nuclear membrane remains intact. Here the nuclear membrane remains intact, but in case of mitosis and meiosis, the nuclear membrane will disappear in prophase and it will reappear in telophase. And the cytoplasm divides irregularly. So in case of mitosis and meiosis, the cytoplasm will divide in a regular manner, regularly. These are all the key features of this amitosis. So this is what the first division, amitosis division. Now where the amitotic cell division will be taking place, which are all the organism which is undergoing this type of cell division? And the first thing will be the prokaryotes. Prokaryotes will undergoing asexual reproduction. See, the main important division that will be the there will be no importance of another cells, which means the prokaryotic cell, just this parent cell gives divided into two, and further this will be divided into two. So that is why we are calling that as a direct cell division, and that will be taking place in the prokaryotes. In the protozoans and aged or degenerating cells is also undergoing amitotic division, and cancer cells that in an abnormal division, so this is also undergoing an amitotic division. So these are all the area where we can find the amitotic cell division. Okay, now what will be the significance? Why the amitotic cell division is important? This is the fastest mode of division when compared to mitosis and meiosis, and it leads to unequal distribution of genetic material. So say for an example, in mitosis, this is a chromosome. Okay, so this is the chromosome, and at the center the spindle fibers is getting attached with the centromere, and finally one chromosome has been divided into two separate chromatids. But as a result, there will be an equal distribution of genetic material. But in case of amitosis, there will be no involvement of spindle fibers, there will be no attachment with the centrosome. As a result, it will not be an equal distribution of genetic material. It is a random division of the genetic material. So that is is why we are saying it leads to unequal distribution of genetic material. Now what we can expect amitosis in the exam? Amitosis is very rare in case of higher eukaryotes. So amitosis is very common in prokaryotes and it is very common in the eukaryotes. I mean common in the prokaryotes as well as the protozoans. So this is what about the amitosis division. Now comes to the mitotic division and the meiotic division. So in mitotic division, so we have know that we have G1 phase of cell cycle, S phase, and G2 phase and M phase. And what we have discussed about this G2 phase? This G2 phase is the preparatory phase for the M phase, and M phase where exactly the cell division is gonna take place. See, in mitosis there are different phases is coming under the mitosis, and where there is a prophase, metaphase, that is pro-metaphase and metaphase. We just remember that there is a prophase, a metaphase, anaphase and telophase. So these are all the phase which is coming under the mitosis. And in case of meiosis, further this meiosis is classified into two phases, that is meiosis 1 and meiosis 2. And in the meiosis 1 there is a prophase 1, metaphase 1, anaphase 1 and telophase 1, and prophase 2, metaphase 2, anaphase 2 and telophase 2. So these are all the phases which is coming under the meiotic stage 1, meiotic cell division 1 and meiotic 2 cell division. We are going to discuss about that now. Just see here mitosis, and this mitosis is also called as a equational division. Why we are saying this particular mitosis as equational division? Let me discuss that. See here, mitosis is a type of cell division. We know that, and in which one parent cell divides into two genetically identical daughter cells maintaining the same chromosomal number. And these are all the phase. See here, one cell is divided into two cells. So that is what it is, a type of cell division, and gives rise to two daughter cells. And whenever the one parent cell is divided into two daughter cells, each daughter cells are having a genetically identical, just as like a mother. And now the mother is having a 46 chromosome number, and each daughter nuclei is also receiving a same equal 46 and 46 chromosome number. That is why we are saying this as an equational division. All the contents from the mother cell is been carried to the two daughter cells exactly the same, right? So this is why we are saying this mitosis as an equational division. Now, where exactly this mitosis is going to take place? This mitosis is taking place in the body cells. That is what the somatic cells, except two cells, that is the germ cell. What is in case of germ cells? In case of men, in case of female, we are calling that as an egg. In case of men, we are calling them as a sperm. So, oogonial mother cell and sperm mother cell. So these are considered as a germ cell, and except this germ cell, all other cells we are calling them as the body cells. And in all these body cells, which type of division is important? Mitotic division is taking place. So this point is very, very important. And in case of plants, again this mitotic cell division is going to take place. And where exactly the mitotic division is taking place? In the meristematic tissues. So as of now, just know that this why we are calling this mitosis as an equational division, and what are all the reasons. Now, where exactly this equational division is taking place in case of animal as well as the plants. Okay, now we are going to discuss about what will be the significance, what will be the importance of this mitotic cell division. Growth. See here, this is a baby, and this is whenever we are taking a baby, the baby is too small. And when we are, just imagine yourself when you are in a baby and you are now. Whenever you are in a baby stage of the baby, you are looking very small, and after that now you are a well grown adult where all the organs have been properly attained a certain growth. And how all these are possible? A small baby without cell division, without cell cycle and cell division, the small baby cannot be converting into a big well grown adult. So for that reason, a cell division is very, very, very important. Now tissue repair, that just now I have explained you in the starting slide. So it is involving in the tissue repair. So whenever I got some damage in my hand, that has to be repaired, and how that damaged cells has been undergoing an apoptosis process, and all this apoptosis is nothing but a cell death. So if any cells are getting damaged, a newer cell has to be developed, newer cell has to be divided. So all these process has been done with the help of the cell division process. Now asexual reproduction, just see here, this mitosis, this is the there will be no see, in case of sperm, it has to be meet with the ovum, and it will be fertilization occurs. As a result of fertilization, zygote, and this zygote gives rise to a new born baby, and this is what a sexual reproduction. But this mitosis, it is asexual reproduction. This cell, to undergoing a division, it doesn't need to mate with any other cell, it doesn't need to mix with any other cell. Just this cell, when it attains certain size, it gets some signal, like what we have discussed in the previous class, the checkpoints, and on all the cyclins and cyclin dependent kinase is the one involving in stimulating the cell division. Now this is considered as an asexual reproduction, and also it maintains a genetic stability. Even though cells is undergoing a millions and millions of cell division, they are maintaining a genetic stability. What this parent what are all the contents is present in the mother, even it will be divided into two cells, the same content has been reflected in the daughter cells also. So that is why we are saying this mitosis is maintaining the genetic stability. So these are all the importance of this mitosis. Now, what are all the phases which is coming under this mitosis? So we can classify this mitosis into two different stages. So in yesterday we have discussed about these two main stages. Just listen to me here. What are all the two main stages of this mitosis? Karyokinesis and cytokinesis. Karyokinesis, the word kinesis refers to division, and the word karyo refers to nucleus. So the division of nucleus is called as a karyokinesis, and there is a kinesis. Again, it is a division, and here it is not a karyo, it is cyto. Cyto, in the sense, it refers to cytoplasm. So the division of cytoplasm is called as a cytokinesis. So the two main stages of mitosis is nothing but karyokinesis and cytokinesis. Karyo means division of nucleus, and cyto means division of cytoplasm. Just look at this picture. In the amitosis process, I have shown you an important picture here. Inside a cell, there is a nucleus. The nucleus is the one undergoing a division first. So you should know karyokinesis is the first stage, and karyokinesis is followed by the cytokinesis. Only after the nucleus has been divided, each nucleus has been surrounded by a cytoplasm, and it will be divided into a different cell. And you should know this statement is very, very important statement. Always the karyokinesis occurs first, followed by the cytokinesis. Karyokinesis followed by the cytokinesis. So these are all the two main stages under the mitosis division, and you know what will be the stages and what is the meaning of it. Now, under the karyokinesis, that is the nuclear division, there will be a different phases, and we are going to see that. Just look at this prophase, very, very important phase of this nuclear division in the mitosis process. See, in the mitosis we have prophase, metaphase, anaphase, and telophase. Four phases, we are calling them as the mitotic phase. And in the mitosis, we can further classify it into two, that is a karyokinesis and the cytokinesis. In the karyokinesis, the first phase will be the prophase. And what are all the events which are taking place under the prophase is the chromatin is condenses to the chromosome, which means chromatin condensation. The chromatin condenses into chromosome. This is very, very important, and this chromatin condensation is not taking place in the amitosis, but it is taking place in the mitosis. And where exactly in the mitosis? That too specifically the prophase. And each chromosome has two sister chromatids. See, this is a chromosome and this is the chromosome. Each chromosome have two chromatid. right? So each chromosome have two sister chromatids. If the sister chromatids present in two different chromosomes, then this is a chromatid and this is a chromatid. We are calling this particular chromatid as non-sister chromatid. So the chromatid which is present in the same chromosome, which is called as a sister chromatid, and the sister chromatids of two different chromosomes, we are calling them as the non-sister chromatid. And centrioles move to opposite poles. Here just look at the picture. Let me explain you about the picture. But know that chromatin condensation is taking place in the prophase, and in each chromosome we are having two sister chromatids, and the centrioles here is moving in opposite direction. Why it has to move opposite direction? Let me explain you. The spindle fibers begins to form. This is where we can expect the question. So this is the spindle fibers begins to form in the prophase itself, and we have studied the spindle fibers is attaching in the centrosome. Centrosome is the portion where two chromatids is getting attached with each other. The spindle fibers is the one by attaching with the centrioles, it has been centrosome, it has been pulling out the two sister chromatids into single chromatids. So this is very important for splitting of for chromosomal segregation. This spindle fibers is very important. The nucleus is getting disappeared. Nucleolus is getting disappeared. So see here. So this prophase is the longest phase of the mitosis. And see here, this is the prophase region. In the prophase, the nucleolus is getting disappeared. So the separation of the centrosome, formation of mitotic spindle, chromatin condensation into chromosome. So all these processes will be taking place in the prophase, and we have studied prophase, metaphase, anaphase, telophase. These are all the four phases coming under the mitotic phase. When compared to all these forces, prophase will be the longest phase of the mitotic phase. Just look at the picture. Yeah. Now just look at the second phase. Again, this is the phase coming under the karyokinesis only. See here, metaphase. In metaphase, see, just look at this picture. See, these are all the chromosomes, and we are having 23 pairs of chromosomes and 46 number of chromosomes. And all these chromosomes will be aligned in the center. And these, just look at here, the centrioles. Centrioles is moving towards the opposite poles in the prophase, and see here, now the spindle fibers secreting the centrioles is secreting the spindle fibers, and all the spindle fibers is getting attached to the kinetochore. Kinetochore is nothing but a structure which is present in the centrosome. And centrosome is nothing but the central point is called as a centrosome. Okay, now all these chromosomes are arranged in the equatorial plate. So this is what an equatorial plate. Right, now the spindle fibers is attached into the kinetochore, and the chromosome shows maximum condensation here. So the chromatin condensation will be taking place in the prophase, and the metaphase, the chromosomes, now it is showing a maximum condensation. And in the exam point of view, in exam, what we can expect from in the cell division? This is very important point. Best stage of karyotyping. When a scientist, whenever you are going to become a scientist, just know that only at the metaphase you can analyze each and every chromosome's function. So the best stage of karyotyping will be the metaphase. So this is very, very important, and this point is important. All the chromosomes has been aligned at the central point, that is at the equatorial plate, and the centrioles in the opposite pole, and they are secreting the spindle fibers, and the spindle fibers is getting attached with the kinetochore, and this kinetochore is nothing but it is a structure which is forming the centrosome. Now the third important phase, another phase which is coming under the karyokinesis, is anaphase. Here, now what are all the chromosomes which is aligned at this equatorial plate? Now it has been segregated into chromatid. That is what we are saying that as a centromere split. Just see here, this is where the chromosome has been aligned in the metaphase. Now one chromosome has been split into two chromatids, and one chromatid is moving at this center, this corner, and one chromatid is moving at the right side. Okay, now sister chromatids are getting separated only in the anaphase, not in the metaphase. In the metaphase they are getting attached with the kinetochore, but in the anaphase is getting split. Daughter chromatids move to opposite poles. This is very important. Now another important characteristic features of this anaphase will be, this will be the shortest phase of the mitosis. Just look at this picture. In this anaphase there is this is a outer surface, there is what, a plasma membrane, and here the two sister chromatids of one chromosome has been split. Now this is the structure which is responsible for the formation of spindle fibers that is arranged at the opposite pole, and for the separation of these two chromatids there will be the, see, after the chromatids has been separated, there is a cleavage furrow has been at the center, which means what? Let's look at the picture. There is a furrow like structure here. So this structure is called as a furrow. This bent like structure is called as a furrow. Why? Because now the nucleus has been started to segregating. The chromosome has been segregated and at now this is the phase under the karyokinesis. The nucleus has been started to divide and it is dividing. Now the karyokinesis has to be followed by the cytokinesis. So for the cytokinesis process, this cleavage furrow is very important for the cytoplasm to divide. Okay. Now in telophase, chromosome decondensation has been taking place. Now the chromosome has been decondensed. Chromosome condensation will be taking place in the prophase and the metaphase, and the decondensation of chromosome will be taking place in the telophase. Now the nuclear membrane will be disappear in prophase and it is reappearing in the telophase. The nucleolus reappears. It is disappear in the prophase and it is reappear in the telophase. Now the spindle fibers appear in the prophase and the metaphase, and also in the anaphase. The spindle fibers is disappearing in the telophase. This is very important. Now just look at this. The cleavage furrow is little more intense. Now as a result of this cleavage furrow, two dotted cell has been completely circled by the cytoplasm. Now these are all the phases. There is prophase, metaphase, anaphase, telophase. All these four phases which is coming under the karyokinesis. Again it is a type of a mitosis. Now this karyokinesis has been completed. It has been followed by the cytokinesis. The cytokinesis is nothing but a division of cytoplasm. The division of cytoplasm. In case of animal cell, just now we have studied there is a cleavage furrow which is an important structure for the cytoplasm to divide. So in case of plant cell, that structure is cell plate, that is called as a phragmoplast. So cell plate formation, that is nothing but a phragmoplast formation, that is an important structure for the cytoplasm to divide. So that process is called as a cytokinesis. So see here, so cytokinesis is centrifugal in plants and centripetal in animals. So this can be expected in the exam. This is very important. Now this is what exactly the cytokinesis. So just to see it, this structure is called as a cleavage furrow. Now each dotted cell has been divided into two different cells and it retains its chromosomal number. So this process is called as a cytokinesis process. Now coming to the meiotic cell division. So this meiotic cell division, it is also called as a reduction division. See what we have discussed, this amitotic division is also called as the direct cell division, and in the mitotic cell division it is also called as an equational division. Why? Because the chromosomal number in the mother cell has been divided properly. The same number has been seen in each of the daughter cells. So that is why it is also called as an equational division. Now why this meiosis is called as a reduction division, we are going to discuss about that. See, the meiosis is a special type of cell division, that is one diploid cell, that is the 46 chromosome, we are saying that as a diploid cell, it has been divided into 4 haploid cell and 4 haploid cell, which means what? Now 46 is considered as a diploid and the haploid means 23 chromosome, and one cell here in meiotic division it will be divided into 4 cells. So just see here, the mitotic cell division is also called as an equational cell division, and the meiotic cell division is also called as a reduction cell division. And in the mitotic cell division, it will be taking place in the somatic cell, but here in the meiotic cell division, it will be taking place in the germ cells. And all the germ cells has been synthesized, and that particular process is called as a gametogenesis. The germ cells are responsible for producing the gametes, and the gamete synthesis process is called as a gametogenesis process. Only these germ cells is the one undergoing which type of cell division? Meiotic cell division, that is the meiotic 1 and meiotic 2 cell division. Right? Now, in the mitotic cell division, one diploid cell gives rise to two diploid cells. This is why we are saying that particular division as an equational division. Now, in case of a meiotic cell division, one diploid cell divides into 4 haploid cells. So, the count has been reduced in each of the daughter cell. That is why we are saying this particular division as a reduction division. And also, you should know one more important point will be that the mitotic cell division, they are having similar genetic, they have been restoring the genetic similarity, identical genetics. That then just see here, in meiosis each are genetically different. So these are all the difference between mitotic division and the meiotic division. We are going to discuss about that further. Now, what will be the importance of this meiotic cell division? They are the one maintaining the chromosome number across the generations. How they are maintaining the chromosome number, we are going to discuss about that. And they are the one introduces the genetic variation, how it is introducing a genetic variation. So, in the question, we can expect that whether the mitosis or meiosis, which is responsible for genetic variation, and you should know that will be meiotic cell division, not mitosis cell division. Now, this is the basis of sexual reproduction, but the mitosis instead it will be asexual reproduction. So, these are all the drastic difference between this mitosis as well as the meiosis. Now, so let us see that what are all the phases which is coming under the meiotic division. Now, as I have mentioned you before, this meiosis stage can be divided into meiosis 1 and meiosis 2, and further this meiosis 1 divided into prophase, metaphase, anaphase and telophase, and meiosis 2 will be categorized in prophase 2, metaphase 2, anaphase 2 and telophase 2. And what are all the significant processes will be taking place in each of the phases. Now, just listen to me. This prophase 1, it is very important for the exam. So, as I have mentioned you before, meiotic 1, further it will be classified into prophase, metaphase, anaphase and telophase. And prophase 1, anaphase 1 and telophase 1 and all these. See, further this prophase 1 will be classified into 5 different types: leptotin, zygotin, pachytin, diplotin, diakinesis. And the first stage will be the leptotin, and this leptotin appears in the thin thread. So here the thin shaped chromosome will be appeared, and that particular stage is called as a leptotin. So in the leptotin phase of the prophase 1, all the chromosome has been appear as thread like structure, and this particular phase is called as the leptotin. Just look at this picture. This is the centromere, and usually the chromosome will be like this. And in case of leptotin, it will be like this. So this is why we are saying the chromatids chromosomes are in a thread-like structure. So that will be taking place in the leptotin. Now, the next phase of the prophase 1 will be the zygotin. So in the zygotin, the pairing will be taking place. Pairing of the homologous chromosome. Homologous chromosome in the sense the structure will be same and the functions is also same of both of the chromosome, that is called as a homologous chromosome. So those chromosome which looks exactly similar in their size and shape, we are calling them as a homologous chromosome. And if the pairing occurs between these two homologous chromosomes, this pairing stage is called as a zygotin stage. Just look at this. This is called as a pairing. The pairing will be occurs in the form of pairing will be occurs with the help of this synaptonemal complex. So the formation of the synaptonemal complex is also taking place in the zygotin, and the pairing of the homologous chromosome will also be taking place in the zygotin. So this is where exactly we can expect a question. And The third phase of the prophase 1 will be the packeting phase. Packeting phase is nothing but where the crossing over is taking place, and this is why we are saying this meiosis phase is responsible for genetic variation. And for this genetic variation, the basic step will be this crossing over, and because of this crossing over, we are saying that the genetic variation will be taking place. Just look at this picture. This is a chromosome and this is another chromosome, and this is a part of this chromosome 1 and this is a part of the chromosome 2, and the chromosome 2 and this is a chromosome 1. The exchange of genetic material from between the chromosome 2 and the chromosome 1. Now this is the genetic part, which is the chromosomal part, which is coming which is a part of the chromosome 2. Now it has been mixed with the chromosome 1, and the part of chromosome will be mixed with the chromosome 2. So the exchange of genetic material between the chromosome 1 and 2, this exchange is called as a crossing over. Consider this as a mother chromosome and this is a father chromosome. Now the chromosome material has been exchanged. So that is why whenever you are looking at you are a mixture of your mother as well as a father. So this is what we are saying, that as a genetic variation. Okay, now crossing over will be taking place in the packeting. This is very important stage, and the exchange of genetic material, we are calling them as a crossing over, and this crossing over will be taking place in the packeting stage, and this is very important for the exam. And also just look at this picture. One of the most important points, see what we have discussed in the zygote phase of the prophase 1. In the zygote phase, the synaptonemal complex has been formed. As a result of the formation of the synaptonemal complex, 2 homologous chromosomes has been paired with each other. This is what with respect to the zygote. Now coming to the packeting, what we have discussed, crossing over has been occurred. So for crossing over to be taking place, 2 chromosomes is getting attached with each other. So this particular attachment of 2 chromosomes, we are calling them as a tetrad. Right? So you should know that tetrad formation will be taking place in the packeting stage. So this is very important. Now, and the fourth phase of the prophase 1 will be the diplotin phase, and the diplotin phase, here the chiasmata visible. The chiasmata is very essential for the formation of the synaptonemal complex. Now here the chiasma will be visible in the diplotin stage. This can also be expected in the exam. The chiasmata is visible in which phase? That will be in the diplotin phase, and this chiasmata is responsible for the separation of homologous chromosome, and for the separation of the pairing of the homologous chromosome will be taking place in the zygote. Now this pairing has to be separate. That separation has been mediated by this chiasmata, and all these features will be taking place in which phase of the prophase? That will be the diplotin. Now the last and the fifth phase of the prophase 1 will be the diakinesis. Now the chiasmata structure has to be disappear. No need of chiasmata anymore. So the terminalization of chiasmata will be in the diakinesis. Now the nuclear membrane disappear in the prophase 1, which means in the packeting, leptotin and zygotin. Now it will be the nuclear membrane will be disappear. So usually the nuclear membrane is disappear in the prophase and it will be reappear in the telophase. Like in the same way, the nuclear membrane has been disappear in the prophase. But more specifically in case of meiosis I, where exactly the nuclear membrane is getting disappeared. So the disappearance of this nuclear membrane will be taking place in the diakinesis, and the terminalization of chiasmata will also be taking place in the diakinesis. This is very important thing. Now, metaphase 1. So in the metaphase 1, it will be the homologous chromosomes aligned at the equator. Isn't it? See here, now all these processes are just similar to the mitosis. In the mitosis also we have discussed about the metaphase, and always the chromosome has been aligned at the center of the cell that is at the equatorial plate. So that is what the homologous chromosome align at the equator. There is a random orientation and independent assortment. So now the centrioles has been arranged at in the astral rays, the shape of the centriole. It will be arranged at the poles of both sides. Now it will be attached with the spindle fibers from the centrioles is getting attached with the centrosome. So this is what are characters of metaphase now. And in the anaphase 1 here, see here the chromosome, homologous chromosome separated. Finally the homologous chromosome is getting separated. See just what we have discussed, the chromosome with both the chromosomes from the mother and the father. Now the chromosome has been separated. Now in the prophase, the chromosome will be, the tetrad has been formed. Both the mother and the father's chromosome has been attached with each other. That is what the tetrad. Now in the metaphase, both the chromosomes has been aligned at the center. Now in the anaphase 1, they have been attached at the both the chromosome after the crossing over. Now the genetic material between the father and mother has been exchanged. After the genetic material has been exchanged, now it has been separated like this. Now the centromeres do not split. What is the difference between mitosis and meiosis? Just look at this picture. This is what the chromosome, and after the division, it will be divided into chromatids in case of mitosis. Now in case of this meiosis, here what we can expect here? This is a chromosome and this is a chromosome tetrad formation. Now the centrioles has been, the spindle fibers has been attached here. Now two chromosomes have been separated like this. Now this chromosome has both mother and father genetic material, and this chromosome is also having mother and the father genetic material. So this is what exactly happening in case of anaphase 1. Here the reduction in chromosomal numbers occurs here. The reduction in chromosome number occurs here in anaphase 1. In the telophase, two haploid nuclei has been formed. The chromosomes still have two chromatids. They are having two chromatids in the telophase 1. Just look at this picture. Next, meiosis 2. In meiosis 1, half of the reduction division has been the chromosome number has been reduced from diploid to the haploid. Now in meiosis 2, in this meiosis 2, it is exactly similar to the mitosis. That is what the stages under the meiosis 2 will be: prophase 2, metaphase 2, anaphase 2 and telophase 2. And the result of this meiosis 2 will be 4 haploid and genetically distant cells. And you should know meiotic 2 division will be exactly similar to the mitosis 1. Now what are all the phases which is seen under the mitosis will be exactly similar to the meiosis 2. Now the difference between mitosis and meiosis, the first important division will be where it is exactly occurred. In case of mitosis, it will be occurring in the somatic cells. In meiosis, it will be occurring in the germ cells. It will be the division of mitosis will be only one, and meiosis meiotic 1 and meiotic 2. That's why two divisions. That is the reduction division is taking place and equational division is also taking place. And as a result of mitosis, 2 daughter cells has been produced, and here 4 daughter cells has been produced. That is 4 haploid. And here the chromosome number has been maintained throughout the division, but here the chromosome number has been reduced. And the crossing over has been absent, and here the crossing over is present. And genetic variation, there will be no genetic variation, and here it will be, the variation will be occurred. Now, so with respect to the cell cycle, all those cell cycles has been discussed in yesterday's class itself. Kindly go through it, and these are all the thing. And now abnormal cell division. So the abnormal cell division, which is nothing but a cancer and non-distinction. Uncontrolled mitosis, which is what we are saying that as a cancerous cell, and where there will be the loss of checkpoint control, there all we can see the uncontrollable cell division, and that particular condition is called as a cancer. And non-disjunction. So we have keep on studied one chromosome has been segregated into two chromatids, and if that segregation is not functioning properly, and that particular condition is called as a non-disjunction, failure of the chromosome separation, and this leads to aneuploidy. Only because of this non-disjunction, Down syndrome we have discussed in the previous class. Down syndrome is the condition which is taking place. Now, we have to discuss about this P53 and P51. See, this P53 is considered as a tumor suppressor cells. Wherever there will be a loss of checkpoints, there we can see the uncontrollable cell division. We can call them as the cancer. And the gene which is responsible for giving the cancer is called as an oncogene, and the gene which is responsible for suppressing the cancer causing gene is called as a tumor suppressor gene. And this P53 is one of the tumor suppressor gene. And what will be the function of this tumor suppressor gene, that is P53? And oncogenic signaling, if there any gene, oncogene means what? Gene which are causing a cancer, the signal will be received by the P53. And if there is any oxidative stress, if there is any hormonal imbalance, physiological process, and if there is any DNA damage, if there is any oxygen supply condition is absence of oxygen condition, that is hypoxia condition, and if there is any nutrient deficiency, in all these conditions the P53 is getting activated. See, I have listed out what are all the conditions. I have listed out what are all the conditions which is going to activate the P53. So these are all the conditions. It results in the activation of the P53, and P53 is nothing but a tumor suppressor gene. Now, this P53, whenever the P53 is getting activated, the cell cycle has to be stopped. And what are all the cells which is not having their nutrient properly, and what are all the conditions said here? If any cells undergoing in any of these conditions, that cell will be undergoing a senescence stage, and that will be done by the P53. And how they are going to done this? See, if there any damage occurs in the cell cycle in any of the cell cycle is getting arrested. This particular cell will not allowed to enter into the cell cycle, and this condition is called as a cell cycle arrest. Now, what happened to that particular cells which is getting arrested by this P53, and that particular cell is undergoing an autophagy condition or apoptosis condition. Apoptosis means programmed cell death. See here, this programmed cell death, that is what an autophagy, and programmed cell death, that is nothing but an apoptosis. And necrosis, necroptosis, these are all the part of the cell death. Now, what about this P21? So, P53 is what we have discussed, tumor suppressor gene, and this is the gene which is responsible for suppressing the gene. Now, what is with respect to this P21? This P21 is the CDK inhibitor. So, in the previous class, we have discussed what is meant by CDK, and CDK is nothing but cyclin dependent kinases, and we have discussed about the cyclin and the cyclin dependent kinases. Only with the presence of the cyclin, the CDK is getting activated. Now, what is the relationship between this P21 and the CDK? P21, it is going to inhibit a CDK. Why it has to inhibit a CDK? We are going to discuss about that. Acts downstream of P53. If P53 is getting activated, P21 is also getting activated. Now, see here, if the cell is losing their control, if the DNA is getting damaged in this particular cell, now P53 is getting activated. Now, P53 is getting, once the P53 is getting activated, the P21 is also getting activated. Now, what is the function of this P21? The CDK inhibitor, it is considered as a CDK inhibitor. Why it has to inhibit a cyclin dependent kinases? So, only with the presence of the cyclins and cyclin dependent kinases, the cell cycle is being continued. Now, if we suppress this cyclin dependent kinases, the cell which is having a DNA damage will not enter into the cell cycle. So, that is why this P21 is getting activated when this P53 is getting activated. Now, just see here. See here, when there is a cyclin CDK7 and cyclin and CDK is there, and the P21 which with the CDK and cyclin. And what is the function of cyclin dependent kinase? To add the phosphate group. Now this phosphate group, that is CDK, is now getting phosphorylating the retinoblastoma, that is RB. Now it is going to activate the transcription factor called E2F. Now this is one, now the P21 is getting activated. Now just look at this picture. If there is a CDK and cyclin, the CDK molecule and cyclin is present. If the cyclin is not present, then the P21 is getting attached with the CDK and cyclin. Now there will be no retinoblastoma and no activation of this E2F factor. Now it results in the inactivate complex. See here. Yes. Now this retinoblastoma, what is the function of this retinoblastoma protein? So this is the one preventing the G1 phase, and it has to enter into the S phase. Phosphorylation inactivates the RB. So see here, if the inactivation of this retinoblastoma, what is exactly is going to happen? Let me explain you detail with the help of this picture. See, what have we here discussed so far? Yes, see here. So this is what P53. If the P53 is getting activated, if it is getting activated, in the sense what? If it is getting activated, there is a cell damage. Consider if any of the damage is occurred in the cell. Okay, if any cell damage occur in the cell, the P53 is getting activated. Now if P53 is getting activated, P21 is also getting activated. If the P21 is getting activated, it is inhibiting the cyclin dependent kinases. If the cyclin dependent kinases is getting inhibited, there will be no cell cycle. Now, so we have studied there will be, if there is a cell damage, so that the P53 is getting activated now. Now the P53 is going to activate the P21. Now what is the function of this P21? Now, what is the function of this P21? This P21 is very important. Now, if the retinoblastoma is very important. If the retinoblastoma is getting phosphorylated, it is considered as an inactive complex. If the retinoblastoma is unphosphorylated, if it is not phosphorylated, it is an inactivate complex. Only then the cell will undergoing a apoptosis process. See here, there is a CDK and cyclin. Now there will be no problem for the CDK. Now the CDK is not getting inhibited by the P21. If the P21 is getting activated, if the P21 is getting activated, if it is activated, it is going to inhibit the CDK. If the CDK is not, it is getting inhibited. Now when the P21 is getting inhibited and inhibiting the CDK, now it is not going to phosphorylate the retinoblastoma. See here, now the P21 is inhibiting the CDK. As the inhibition of CDK is happening here, the CDK molecule is not going to phosphorylating the retinoblastoma. If the retinoblastoma is not getting phosphorylated, then it is called as an inactivate complex. Then it is not enter into the cell cycle. Now just see here. Here the CDK is normally functioning. Now the CDK is getting activating the phosphorylation. Now this phosphorylation is also adding the phosphate group to the retinoblastoma. Now it is getting activated. Now it is continuing with the cell cycle process. Now these are all the one-liners, just have a look at it. Okay, now coming to the first question, during which phase of the meiosis does the synaptonemal complex completely disappear? Which chiasmata persist? So which stage that the meiosis have the synaptonymal complex completely disappears. So we have studied synaptonymal complex forms in the zygote and it will be disappears in the diplotin stage. Now, the reduction in the chromosome number during the meiosis occurs due to the reduction. Chromosomal number is getting reduced during the meiosis occurs due to which stage? So, whenever there is a separation of the homologous chromosome, at this stage the chromosome number is getting reduced. Now, which of the following statement is incorrect regarding the metaphase of mitosis? See here, incorrect phase regarding the metaphase of the mitosis shows a maximum condensation. Of course, it is a right statement. Sister chromatids are identical and the centromere split at this stage and the chromosome aligned at the equatorial plate. So the answer for this question will be it's split at this stage. The chromosome is not splitting at the metaphase stage. Non-disjunction during meiosis I differs from the meiosis II because in meiosis I it leads to, just see here, non-disjunction is during meiosis I, it differs from the meiotic II. Why it is differing? Equal distribution of chromatids, all gametes are being abnormal, half normal, half abnormal gametes, and absence of aneuploidy. Why the reason for this? All gametes here are being abnormal. Okay, so if the non-disjunction is not proper in the meiosis I, it leads to abnormal gametes. Now, which checkpoint of the cell cycle ensures that all the chromosomes are properly attached to the spindle fiber before cell progression, and which is that will be the spindle M checkpoint, that is spindle or the M checkpoint, mitotic checkpoint. Chiasmata represent sites of spindle attachment, points of the synapses, physical manifestation of crossing over, and regions of the centromeric fusion, and this chiasmata represents the physical manifestation of the crossing over. And correct matches, you have to identify the correct match. Leptot internalization of chiasmata, no, it is wrong. Zygote crossing over, no, it is wrong. Packet in stage are recombinant nodules, and diplotin synaptonymal complex formation. So the answer will be option C. Cytokinesis in plant cells differ from the animal cells because the plant cells, they are having centripetal division with cleavage for row. No, it is wrong. This is with respect to the animal cell. And centrifugal cell plate formation, actin-myosin ring formation, no. Absence of Golgi complex, no. The answer will be centrifugal cell plate formation. So which protein is correctly termed as a guardian of the genome due to its role in halting the cell cycle upon cell damage? That will be the P53. Just now we have studied. So if crossing over fails to occur during meiosis, the most direct consequences will be, if there will be no crossing over, there will be reduced genetic variation. So that's all about the cell division. So we have completed the cell biology chapter thoroughly. Okay, so kindly go through it. We'll meet you in the next class.